So we have this picture of the cell cycle as this inexorable cycling through phases where one cyclin comes, activates another cyclin, and then it goes away, the other cyclin progresses to the next phase, so on and so on forever and ever. Of course, this is a vast oversimplification. What if the extracellular environment is not favorable? Those cells tend to just stay in G1, or maybe we could call it G0. What if there's DNA damage? In that case, the plan is to pause or die and not progress through this cell cycle. Some more complicated phenomena are associated with the number of chromosomes or the number of genomes. And this is an interesting phenomenon here where the M-phase cyclin is actually designed to prevent something that would happen in the next S phase. So we'll talk about that real briefly by the time we're done. We're not going to talk too much about chromosome separation in this class. But what are these arrows with the solid lines and not the points? What is going on in controlling the cyclin CDK system such that it isn't absolutely inevitable or inexorable? So that's the subject of this video. And there are a number of mechanisms that we can superimpose upon the levels of cyclin as being the controls of cyclin CDK activity. Maybe the main one, certainly the most easy to describe, is phosphorylation. So the way I've described things so far is if you have cyclin and CDK, then you have active CDK activity. That's an oversimplification because there's an additional layer of good housekeeping seal of approval that has to go on, and that is there has to be the appropriate panel of phosphates on the CDK in order to get activity in the presence of cyclin. So that if you have cyclin, you may not necessarily have CDK activity until those phosphates are laid on correctly. So there's a relatively complicated panel of enzymes that operate on the CDKs, one of which is the so-called CAK, CDK activating kinase, which transfers a single phosphate to a particular region of the CDK, and that leaves you with an active cyclin CDK. But there are other phosphorylation mechanisms that are sort of superimposed on that phosphorylation event. In particular, there is the equilibrium between a kinase and a phosphatase. It's kind of our standard story, the yin and yang of kinase phosphatase in one single reaction. In this case, the weak kinase phosphorylates, but in this case it inhibits because it adds an additional phosphate beyond that one that was required to activate. So in order to make an active CDK out of this, you have to remove that phosphate and not that one. So this requires another enzyme, CDC25 phosphatase, that reverses the inactivation provided by the weak kinase in order to get an active CDK. So if you sort of add those levels of regulation to this cycle, you can sort of see how well maybe these enzymes, the various kinases and phosphatases, can be tied into the level of DNA damage, some sort of proofreading function, and relatively quickly modulate the activities of these cyclin CDKs, and that's exactly how it works. Okay, there is another superimposed layer of regulation that is especially important in the case of things like DNA damage that I need to address before getting any more specific, and that has to do with the collection of inhibitors in this figure, exemplified by one of many such inhibitors, often indicated by a little p and then a number that would include something like P16, P21, and in this case P27. So these are inhibitors of the cyclin CDKs, and they do that by physical interaction with what would otherwise be an active form of that kinase. And as a result of the association between that those two subunits and the inhibitor, you get an inactive one, and this is a reversible reaction. And the way to think about this is that now the progression through the cycle the cell cycle, whatever phase it is, whichever cyclin this is going to depend on the relative levels of this versus this. So that if the level of cyclin CDKs exceeds the level of P27 or the other Ps or the inhibitors of kinase, the so-called INKs, then you get progression through the cell cycle. But if not, you don't. So that life is a balance between those two concentrations, as it turns out. So we have the balance between kinase and phosphatase, and we have the balance between cyclin CDKs and the inhibitors of those enzymes that all weigh in terms of the fate of the cell. Okay, now I've already mentioned proteolysis, but I've mentioned it in a sort of limited context. So this figure 1715 expands that concept. So now that we have the concept of inhibitors, we can talk about it. First of all, this really should be control of cyclin CDKs by APCC, but never mind. So what's being shown here is the active M-phase cyclin CDK complex, and it goes without saying that it has the appropriate phosphorylation panel on it and is not bound by an inhibitor. So it's doing the work of mitosis, never mind what that is. But when its time has come, sort of halfway through mitosis really, then you have an active complex that is essentially an E3. So it causes the ubiquitin conjugating enzymes to accumulate and lay down the polyubiquitin chain that is recognized by the proteasome. So then we get the degradation of the cyclin. The CDK is still there, but it's completely inactive because there's no cognate cyclin to activate it. So that essentially terminates the cell cycle, really. It puts an end to the final phases of the M phase and licenses the progression into the next G1 phase. So that's the standard proteolytic control that I talked about in the first video. But because there are inhibitors, there are other ways of using proteolysis to control, and one of a very common one is exemplified by this SCF, which is really just a ubiquitin proteasome pathway, a specific one. But in this case, we have a CDK inhibitor protein that, if it's present, it will associate with cyclin CDKs and inhibit them. So in order to proceed through whatever phase that is, and it's going to be S in the case of the SCF, then this has got to be gotten rid of. So I talked about the balance between the concentration of the inhibitor proteins and the cyclin CDKs. That balance is contributed to by proteolytic processes. As a result of this very, very elaborate E3 recognizing the phosphorylated form of the inhibitor protein, leading to its polyubiquitination and degradation by the proteasome. So you remove the inhibitor by proteolysis, you are left with the active cyclin CDK, and then you proceed through the next stages of the cell cycle.